In Lab 5, we're going to be looking at how we can observe bacteria via the microscope using both direct staining and indirect staining. And we're also going to use this lab to help us learn how to prepare a slide for staining. So again, this is another lab that has a fair number of videos to watch on YouTube. And this first one here is quite important: how to prepare a slide for staining when using bacteria from an agar culture. We're going to be doing staining in many labs during the semester, and in each case we need to learn how to properly prepare the slide. There's a short video on how to chemically fix a microscope slide using methanol, but we're not going to use the chemical method, so it's there more for information. We do need to know how to heat fix a microscope slide. Once we prepare the slide for staining, then we have to heat fix it so the bacteria are not washed off the slide. There's a video on how to make a direct stain and how to make an indirect stain, and then back from Lab 1, reviewing how to focus using oil immersion microscopy. So again, make sure you watch that video, video 6 again, because you've probably forgotten some of the steps involved in focusing using oil immersion, and we'll need to use those today. And of course, don't forget to look at the performance objectives at the end of the lab, as we always do, because that's what you'll be quizzed on, both the theoretical ones and the ones under results that are practical questions. And the practical questions here are basically recognize a direct stain versus an indirect stain. And then anytime we're looking at organisms through the microscope, you need to be able to tell me the shape, arrangement, and or form, which we learned back in Lab 1. And of course, once you've viewed the videos, make sure you do your pre-lab quiz on Respondus. Remember, that has to be done at least 2 hours before the start of your lab. So let's get into the overview of today's lab. Now, in our lab, we have 2 ways we can observe bacterial morphology, which refers to their form and their structure. We can look at living unstained organisms, what we call a wet mount, where we just put a broth culture or a liquid suspension under a microscope and look at it. Or we can look at killed stained organisms. Now, of course, as we saw when we looked at the yeast back in Lab 1, unstained organisms are pretty much colorless with very little contrast. So they're pretty hard to see if they're unstained. So normally we stain organisms for several reasons. We stain them so that there's greater contrast between the organism and the background. In some cases, we stain them to differentiate morphological types. By staining them, we can determine the shape more readily, the arrangement, the form. If we do a gram stain, we can determine if it's gram positive or gram negative. And we can also stain to observe certain structures. In the upcoming labs, we're going to be doing flagella stains, capsule stains, and endospore stains to observe those bacterial structures. But no matter what we're doing, how we're doing it and what we're doing, we need to learn first how to fix the microorganism to the slide so it doesn't wash off. We're going to be putting the bacteria, mixing them with a drop of water on the slide, and then we're going to be staining them with liquid dye and then washing the excess dye off with water. And all of that, of course, would cause the bacteria to wash off the slide as well if we didn't have the bacteria stuck to the microscope slide. So the reason for fixation, of course, is so the organisms won't be washed off during staining. And we can either use heat fixation or chemical fixation. And again, we have a video on both of those. Now, what we're going to be using is heat fixation because it's a little faster to use. And we do have limited times in our hour and 20-minute labs. So we heat fix them by holding the bottom of the microscope slide against the opening of your Bacto incinerator for 10 seconds. The heat in turn coagulates the proteins in the bacteria, causing them to stick to the microscope slide so they're not washed off. Now we can also use chemical fixation, where we take an air-dried smear of the organism and we cover it with 95% methanol. Let that sit a couple minutes. Pour off the excess methanol and let it air dry. And in this case, the methanol causes proteins to stick to the slide. Now, this actually causes less damage to the bacteria and is a little better at sticking microorganisms to the slide, but the methanol does have to be very fresh, and it does take longer for this to do this fixation procedure. So we're going to be using the heat fixation in our labs, the first one. Now, as I mentioned, one of the most important videos, the first one, is how to prepare a slide for staining when using bacteria from an agar culture, your video 1. So to learn that, go to the video first. Now on this page here, on the introductory page, I've gone through the steps kind of in writing step by step. But again, you're much better off following the video because that shows you kind of in real time what you're doing. Now, most labs, we are going to take the bacteria off of an agar plate in order to stain them rather than out of a broth culture. So keep in mind, if they're growing on an agar plate, all the bacteria are growing stuck to one another, piled up on top of one another. And you can't see shapes and arrangements. You can't get reliable gram stains if the bacteria just piled on top of one another. So we need to separate the bacteria in a drop of water on the slide so we can see individual shapes, arrangements, and such. Now, since we're often staining several bacteria during one lab period, we're going to put a small piece of tape on one end of the slide and label that with the initials of the bacteria. So remember to do that so you know which bacterium is on which slide when you go to look at it under the microscope. And also that reminds you which side of the slide is up. The tape is on the top of the slide where the bacteria are. Because if you accidentally turn the slide upside down somewhere during staining or put it on the microscope upside down, it won't focus. And I've seen people spend 20 minutes trying to find stuff on a slide that's turned upside down. You can't see them if the slide's upside down. Now, since the bacteria are growing en masse on colonies, we need to add a small drop of deionized water. And we have dropper bottles with that. I normally write half of a normal size drop, half of what would normally drop out if you let a drop fall out of the dropper bottle. Even a half is approaching a little large. A quarter might even be better. So figure 2, and again, you see this better on the videos, something like this on the left would be way too much water. That could take over an hour to air dry, and you have to just sit there and wait for it to air dry before you can continue staining. So that's more close to the correct amount of water you want. When that drop of water is on the slide, it should be no larger than the diameter of a green garden pea, like the peas you eat. Anything bigger than that's too much water. And if you get too much water on, you can just blot it off and try again, put a smaller drop on. But the point is here is that the smaller the drop of water, the faster it's going to air dry, the sooner you're done. Then using your sterile inoculating loop, remove a small amount of the culture from the agar surface, and we're going to just touch it to the drop of water several times until the water just becomes visibly cloudy. Now, there's a bit of an art to this, and it's really critical you learn how much to put on. In my labs, I'll be demonstrating how much to put on the slide so you can see that as you get ready to start doing it. But the point is that normally I just take the loop and push it into the water once, check it and see if it looks partially cloudy. If not, push it in a second time. If not, push it in a third time. Sometimes one little push and it's perfectly fine. Sometimes you may have to push the loop in several times to get enough to come off where the water looks just visibly cloudy. So you have to kind of watch that carefully. If we look at figure 3 here, here's a drop of water. Now the drop's a little bit bigger than it should be, but that's I put that large so you can actually see that we don't want to get this whole drop looking like this. That would be way too many bacteria. But when we see there's definitely some bacteria in the drop of water, that's plenty. Then you want to burn the remaining bacteria off the loop. Because remember, if you put too much bacteria in the water, they're still going to be piled on top of one another. You won't be able to tell the shape or the arrangement or the form very well. And if it's a gram stain, you won't be able to get reliable gram staining if they're piled on top of one another. Now, once you've added the bacteria to the drop of water and you've incinerated the rest of the bacteria off the loop, then make sure the loop cools for 10 or 15 seconds, because if you touch that hot loop to that drop of water, the water is going to splatter all over you. And then once the loop's cool, spread the suspension over the slide to get a thin film. So once you spread it out, as you see here, it should be covering most of the slide. You have the right size drop of water, that should cover pretty much 3/4 of the slide when you spread it out. And the more you spread it out, the faster it air dries and the better you separate the bacteria so you can see individual shapes and arrangements. Then, of course, once you've spread it out, sterilize the loop again since there's bacteria on it. Then finally, the patient part. You have to wait for that to completely air dry. You have to air dry it before you can heat fix it. And again, with a small drop of water spread out really well, it might air dry in 2 or 3 minutes time. If you had too much water, it might take 10 or 15 minutes to air dry. So once it is air dried, as we see in figure 5, it should look like a thin powdery film. You should be able to see through it, but you should be able to see with the naked eye that there's bacteria dried on the slide. So it should be a visible thin bacterial film, but one that you can see through. Then we're going to heat fix the slide rather than chemically fix it. So you're going to pick up the air dried slide using the clothespins we have for that, and hold the bottom of the slide against the opening of your Bacto incinerator for 10 seconds. Figure 6 shows that. Again, the video shows this better. Now, rather than forceps, we're going to use a wooden clothespin. That actually holds the slide a little bit better. But remember, the bacteria and your label are on top of the slide, on this side over here. And you're going to hold it right against the opening. You can just let the slide rest on those 2 screws there and do that for 10 seconds. And that will heat fix the bacteria. So that's the procedure for preparing a slide if the bacteria come off an agar medium. Now, if the bacteria come from a broth culture, they're already in a liquid, and they're separated in the liquid, so we don't have to add water. Again, we would label a piece of tape so we know which organism's on the slide, which side's up. Usually, if we're taking it out of a broth culture, we draw a circle with a Sharpie about the size of a nickel on the bottom of the slide, so we know where we're adding the bacteria, in this case to the circular area, but the circle's on the bottom of the slide. Then use your inoculating loop, place 1 to 2 loops of the culture within the circle on the top of the slide. And again, don't use water because the bacteria are already in a liquid. So again, you use your loop to add the bacteria to where the circle are and spread them out over the circle, 2 to 3 loopfuls maybe. Then you have to wait for it to completely air dry, heat fix by holding the slide bacteria side up for 10 seconds against the micro incinerator. So that's the only real difference there if it comes from a broth culture or from an agar plate. But most of the time we're going to take it from an agar plate. And every time I give you a new staining procedure in the next several labs, I'll be repeating this whole fixation procedure for you to follow. Now, once the slide has been heat fixed, we're ready to stain it. So we have to know a little bit about stains. Stains are generally salts in which one of the ions is colored. And there are 2 basic groups or types of stains called basic dyes and acidic dyes, depending on where the color is. If the color portion of the dye is in the positively charged ion, then we call that a basic dye. the color is in the positive ion. And most of the dyes we use are basic dyes like methylene blue, crystal violet, and safranin. On the other hand, if the color portion of the dye is in the negatively charged ion, we call it an acidic dye. Acidic means the color is in the negative ion, like the nigrosin we use for indirect staining. And then we have a ground rule. Because of their chemical nature, the fact that bacteria contain DNA and a lot of proteins, the cytoplasm of all bacterial cells has a slight negative charge if we're growing them at a near neutral pH. So the bacterium has a negative charge. If we use a basic dye where the color is in the positive ion, then the positively charged color portion of the basic dye ionically is attracted to the negatively charged bacterial cytoplasm because opposite charges attract, and that holds the dye inside the organism and on the organism. So the bacteria are stained, but the excess dye washes off during washing. And we call that a direct stain because we're directly staining the bacteria. Or as we see in figure 1 here, all bacteria have a slight negative charge. In a basic dye, the color portion is in the positive ion. So for example, in safranin, a red dye that we use, the color portion is in the positive ion, so the positive dye is attracted to the negative charge of the bacterium. Opposite charges attract, and the dye is ionically held on and in the bacteria, so that when we wash the slide, we can wash the dye off the glass because it has no charge, but the bacteria hold the dye. And we call that a direct stain because we're directly staining the bacteria. And that's because we're using a basic dye with the color in the positive ion. Now, an acidic dye, the color is in the negative ion. And so if we use an acidic dye, the color portion's in the negative ion. The bacteria are negatively charged, and like charges repel. So in this case, we let the dye dry on the slide, and the dye forms a deposit around the organism. So we're staining, in this case, the background, not the organism. The organism's colorless. So if we go back to figure 1, we take the same bacterium, has a slight negative charge. We use an acidic dye like nigrosin, where the color's in the negative ion. Like charges repel, so the bacterium repels the dye. So in this case, we don't wash the slide because if we did, there'd be nothing on it. We wouldn't see anything. We'd wash off the dye. In this case, we let the dye dry on the slide, and as we see, it forms a deposit around the organism. And so we call that an indirect stain because we're only indirectly staining the organism. We're actually staining the background. Now, we don't use indirect stains very often, but they are very nice for looking at the different arrangements of cocci. You don't have the dye stuck between the various cocci in the arrangement. You can see them quite clearly. So we will look at one indirect stain today just to get a feel for that, but mostly we'll be doing direct staining. When we do learn how to do gram stains, those are actually direct stains. So part B of the lab deals with direct staining using basic dyes. Because again, remember the dye is in the positive ion of the color is in the positive ion of the dye. The bacteria have a negative charge, opposite charges attract, so we directly stain the bacteria. And you're going to be staining individually the 2 pure cultures you obtained from lab 3. So remember, in lab 3, you isolated and eventually identified that you had one of 2 gram positives, either Staphylococcus epidermidis or Micrococcus luteus. And you're going to make a direct stain of whichever gram positive you have from lab 3. Now remember, Staphylococcus epidermidis, the genus Staphylococcus is a giveaway. It's obviously a coccus shape, and it's a Staphylococcus arrangement since it's named after its arrangement. So you'd be looking for, of course, cocci arranged in irregular grape-like clusters. That is a Staphylococcus arrangement. On the other hand, if you isolated Micrococcus luteus from lab 3, Again, as a genus name tells you, it's a coccus, but this is a coccus that forms either a tetrad or sarcina arrangement. So again, you're going to be looking for cocci arranged in squares of 4. And remember, since it's hard to tell a tetrad from a sarcina, a square of 4 from a cube of 8, we just say tetrad or sarcina. So again, don't get confused and call this a staphylococcus when you look at it. If it was a staphylococcus, in fact, it would be named staphylococcus. This is micrococcus. That's a clump. Remember, every slide has clumps. You have to look at where the bacteria isolated, and here you see lots of nice little groups of 4, and even the clumps are made up of groups of 4. So that would be a coccus with a tetrad or sarcina arrangement you'll be looking for. And your second direct stain will be of whichever gram-negative you isolated from lab 3. You identified and isolated either Escherichia coli, Pseudomonas aeruginosa, or Klebsiella aerogenes, which used to be called Enterobacter aerogenes. And all 3 of these are small bacilli. But again, you want to make sure you put on the right amount of bacteria so you can see isolated bacteria to show me, so you can see individual bacteria to determine shapes, arrangements, not where they're all piled on top of one another. So there's our 3 gram negatives, and you'll have one of those 3 to look at today. So your main goal today is to make 2 direct stains of your 2 bacteria from lab 3. The gram-positive you isolated and the gram-negative you isolated. And of course, when you show them to me, I'll be asking you the shape and or arrangement. So you might start by doing a direct stain of whichever gram-negative you isolated, Escherichia coli, Pseudomonas aeruginosa, or Klebsiella aerogenes. Now again, you go the first 8 steps. First 7 steps actually here are how you prepare the slide that we went over previously and you saw in the video. So remember, after you label the slide, put a small drop of water, no bigger than the diameter of a garden pea, half of a normal sized drop on the slide. Scrape off a small amount of the bacteria. Just touch the loop to the slide until you see some visible cloudiness in the water. Then incinerate the remaining bacteria from the loop so you don't put too much on the slide. After the loop cools, spread that out as thin as you can over the side to get a thin film. Let that completely air dry till it looks like a thin powdery film, as we saw here. And then hold the slide bacteria facing you over the opening of the microincinerator for 10 seconds to heat fix. Then you're ready to stain it. So this case, you're going to stain it with safranin for 1 minute. Now in a direct stain, it doesn't really matter what dye you use. You just want to see the organism, and it'll stain whatever color the dye is. But one of the things I'm looking at here is that you learn to follow directions so that you know which dye I've said to use on which organism. And again, that's mainly just to see if you're following directions. So we're going to use safranin on the E. coli or Pseudomonas or Klebsiella. So that means you cover the smear with the safranin. You add enough safranin to cover the entire smear. Try not to get it on the taped area. Now we use the larger squeeze bottles rather than the dropper bottles that we used to use. But again, you want to add enough to cover most of the smear. Try to keep the tape dry though. Leave that on 1 minute. Then you're going to pick up the slide by one end, hold it at an angle over the staining tray, using the wash bottles of deionized water on the bench tops. Gently wash off the excess safranin from the slide. So as we see in 7B, we pick up the slide, hold it at an angle. Now when you wash it, you want to direct the water above the smear and let it run down over the smear. And you're going to be watching the bottom of the slide. Keep washing until the liquid coming off the bottom looks relatively clear. Don't look at the smear. You don't want to wash it till the smear is decolorized, or you might be washing off the bacteria. We're just trying to wash the bacteria off the glass, not off the bacteria. So continue to do that until the liquid looks relatively clear at the bottom. Then you can be sure it's pretty well washed. Then at that point, take the slide and put it in a book of blotting papers, or bibulous paper as it's also called, to blot it dry. Figure 8 shows that. And again, we showed you that in the video. Put it all the way in. And then once the slide is in, rub it gently with your fingers, very gently. Don't push down harder, you might break the slide, and then you have to start all over again. So we want to blot off the water because your oil is going to go directly on the smear. And oil and water don't mix, so the slide has to be completely dry before you add the oil. So put it in, rub it gently, move it around 2 or 3 times until you've blotted off all the excess water. At that point, you can put your drop of oil on the slide and observe using oil immersion microscopy. And remember, before this lab starts, review the focusing video back from lab 1, or review your focusing steps from lab 1. And once you have the slide in focus and you've moved it to a good area where you can actually see the shape and arrangement, then you can call me over and tell me what the shape and arrangement is. And of course, remember, make use of the lab book. The lab book is telling you what each one of these looks like, and you have pictures of them to look at before lab. So you just need to find an area that looks like that. Now, eventually, of course, you'll have to be able to tell me shapes, arrangements, and forms without knowing what it's supposed to be. You'll need to be able to recognize those. Then you're going to make your second direct stain using the gram-positive you isolated, either Micrococcus luteus or Staphylococcus epidermidis. So again, the first 7 steps review how you fix the slide, how you prepare the slide for staining and heat fix it. Once you've heat fixed it, you're going to stain this one with crystal violet for 1 minute rather than saffronin. After 1 minute, pick the slide up at an angle, wash off the excess crystal violet, put it in your book of blotting paper, blot it completely dry, add your drop of oil, and observe using oil immersion microscopy. And again, once you get it into focus and you have a good area where you can actually see the shape and arrangement, then you can call me over and you'll get marked off for that slide. Now, if time permits, you're going to make a third slide showing normal microbiota and the epithelial cells in your mouth. So in this case, you're going to take a sterile cotton swab and vigorously scrape the inside of your mouth and gums. Then rub the swab all over the top of a clean slide. You don't have to use water, your saliva will take care of that. So just spread the swab over the surface of the slide, then let it completely air dry. Once it's air dried, heat fix for 10 minutes. Remember, the swabs are going to go in the designated container that says swabs and pipettes only. Don't throw them in the trash. You're going to stain the mouth smear, if you get to do it, with crystal violet for 30 seconds. Then pick up the slide, wash off the excess crystal violet, put in your book of blotting paper and blot dry, and observe using oil immersion microscopy. Now, you might want to look for epithelial cells first using your 10x objective. Get them in the very center of your field of view, and then add your oil and keep switching lenses until your oil immersion lens is in place, and then you can look at your normal microbiota on your epithelial cells. Now, when you're all done with lab today, we need to be careful to dispose of the dye and the wash water carefully. These are all, we have to follow OSHA regulations for getting rid of these stains and this water. So when the lab's all done, take your staining tray that you use to rinse everything in. Carefully pour the used dye from your staining tray into the designated drains located at the 3 lab benches where you're working. Those don't connect to any sewer system. They have pipes that lead to a large carboy at the end of the lab, and that collects all the waste liquid, and then we pay to have that disposed of. So none of that gets into the bay from those 3 lab benches. Make sure you don't pour the used dye down the sinks we use for hand washing, because of course that connects to the college's sewer system, and that water can eventually get into the bay. Now, the procedure for doing an indirect stain with an acidic dye is the next one. And your lab instructor may or may not have you do this one. If we are doing traditional labs, I usually concentrate on the 2 direct stains, because that's the procedure you need to know for all the gram stains we'll be doing during the course. And I'd rather get practice on that, since we won't use an indirect stain after today, but your lab instructor may like you to do the indirect stain. So let me go through the procedure for that. Now I'm going to show you a demonstration of this slide for you to recognize on a practical, rather than have you do the staining procedure yourself. But here's the staining procedure for the indirect stain. I normally have people do an indirect stain of Micrococcus luteus, just because it's more dramatic looking than the Staphylococcus when you see it. When you get a good indirect stain, it kind of pops out like Van Gogh's Starry Night. So in this case, we start out by putting a small drop of the acidic dye nigrosin towards one end of a microscope slide. And we're going to actually add the bacteria using our inoculating loop to the dye and mix the bacteria in with the dye. Then we sterilize the loop. We can set it down. We use the edge of another slide to spread the mixture containing the bacteria and the dye with varying pressure across the slide, so we get alternating light and dark areas. So when we're all done taking the slide, normally we just kind of hold an angle and just keep drag and tap down the slide. And as you do that, you get these alternating light and dark areas. And we need the right thickness to see the bacteria. Where it looks dark here, you won't even be able to see through the dye because it's so thick. If we get out where it's too light, there won't be enough contrast, but there'll be areas here that are in between that give us perfect contrast to see the organisms appearing clear against a dark background, as we would expect on an indirect stain. So remember, on an indirect stain, we're staining the background, not the bacteria. Now, once you've spread the dye out over the slide with the second slide alternating light and dark areas, we let it completely air dry. We don't have to heat fix this because we don't wash it. We don't wash it because if we wash it, we wouldn't see anything. The dye is we want on the slide this time. If we wash the slide, we'd wash all the dye off. And then you can put a drop of oil on that and observe using oil immersion microscopy. And again, remember, at the end of the lab, make sure you pour any used dye from your staining tray down the designated drains in the 3 lab benches, but not down the sinks where we do our hand washing. And then you can record your results here. And again, I've shown you pictures of what they should look like. The Staphylococcus epidermidis, of course, in a direct stain with crystal violet. Micrococcus luteus in a direct stain with crystal violet. And then all 3 of your gram negatives basically look the same. They're going to be gram negative bacilli, staining pink because we use safranin. If you get a chance to do the direct stain of the epithelial cell, showing you your normal microbiota, this is what that looks like. This is an epithelial cell under 1,000 magnification. This is the nucleus. And these are the microbiota bacteria that live on that epithelial cell. So that's a stratified squamous epithelial cell from the mouth with the microbiota on it. And finally, make sure you look at the demonstration of the indirect stain, or do the indirect stain, depending on what your instructor asks. This is an indirect stain of Micrococcus luteus. So notice on the indirect stain, we can see the arrangements of cocci very clearly. The 4s come out very nicely on this indirect stain. We're staining the background and not the organism. So of course that would be a coccus with a tetrad or sarcina arrangement. And of course, don't forget to learn your performance objectives. That's what you'll be quizzed on for Lab 5. And of course, as always, there's a self-quiz with answers at the end. So that's direct staining, indirect staining, and two very important techniques: how to prepare a slide for staining when bacteria are coming off an agar plate, and how to heat fix the slide properly for viewing.